3.
Hello, I'm Leire Baptista, and we're going to present in this project molecular and cellular mechanisms of metastases. Tumor metastasis is a multi-stage process during which malignant cells spread from the primary tumor to discontinuous organs. It involves arrest and growth in different microenvironments which are treated clinically with different strategies depending on the tumor histotype and metastatic location. Non-malignant cells under steady-state conditions proliferate as needed to replace themselves. They age or become injured. However, this can go awry, resulting in controlled proliferating of tumor formation, which can be benign or malignant. Malignant tumors rarely encapsulate. They grow rapidly and invade regional tissues. But benign tumors are slow growing, they have a fibrous capsule, they are non-invasive, and morphologically they resemble their cellular precursor. Now we're going to talk about pathogenesis of metastases. Host-tumor cellular interactions can vary between patients, but the steps or events required for metastasis are the same for all tumors. First, they develop in a vascular network, and they need to evade the host immune response. After, they have to respond to organ-specific factors that influence growth. Once they do, the cell can invade the host stroma, penetrate the blood vessels, and enter the circulation, and then they produce secondary metastases. Only a few cells in a primary tumor are believed to be able to give rise to a metastasis, which is a result of the elimination of circulating tumor cells that fail to follow all the steps in the metastatic process. The presence of tumor cells in the circulation does not predict that metastasis will occur, as most of the tumor cells can enter in the bloodstream and are rapidly eliminated. The entry of tumor cells into the circulation is very common. The process of tumor initiation, progression, and metastasis is rapid, and tumor growth rates are generally slow. What's more, 3/4 of the tumor life history has occurred before the diagnosis, and metastasis can also occur prior to diagnosis. Now we're going to talk about epithelial mesenchymal transitions. Multiple molecules contribute to the metastasis process, and some of them are growth factors, cytokines and chemokines, pro-angiogenic factors, extracellular matrix remodeling molecules, and transcription factors. Epithelial mesenchymal transitions are suggested to result in the loss of epithelial properties, including cell-to-cell adhesion and vessel-apical polarity. EMT, or epithelial mesenchymal transitions, have also been associated with the loss of expressions, or mislocalization of proteins involved in the formation of cell-to-cell junctions, such as E-cadherins, sonodaoccludins, or clotting, and the gain of mesenchymal protein expressions. Thus, a depression in E-cadherin expressions is frequently observed during cancer progression, and solid cancers are associated with the loss of the downregularity of E-cadherin expression.
Hello, I'm Javi, and we're going to talk about the probability of metastasis. Noah suggests that acquired genetic variability, developing chronic tumors and selection pressure, and resulting emergence of the new tumor cell variants that display increased malignancy. This hypothesis suggests that tumor progression toward malignancy is accompanied by powerful genetic instability of the progression cells. To test that hypothesis, Sifon and Fiedler measured the mutation of perimetastatic and non-metastatic clone lines isolated from 4 different murine tumors. They found that highly metastatic cells were phenotypically less stealth than their benign counterparts. This study suggests that metastatic tumor populations have a greater likelihood of cells undergoing rapid phenotypic diversification. This genetic instability, coupled with Darwinian selection, may result in population resistant to normal homeostatic growth controls, chemotherapeutic intervention, immune attack, and environmental restraints. The metastasis origin is highly associated with the E-coloring-catenin complex. E-coloring-catenin complex, an organizer of epithelial structure and function, is commonly disturbed in invasive cancer. E-coloring, a transmembrane protein with an extracellular and intracellular domain, is one of the key players involved in cell-to-cell adhesion. The function of E-coloring-catenin complex is believed to be dependent on intracellular catenins. The ability of E-coloring to decrease cell growth and invasiveness of human carcinoma cells and to suppress the development of osteolytic bone metastases. When E-coloring and alpha, beta, and gamma catenins were analyzed as one group, a significant association was seen between reduction in immunoreactivity of at least one of these four proteins and the presence of metastases. This data indicate that if one of these proteins is downregulated, the function of the other proteins is suppressing tumors and metastasis is altered. On the other hand, specific E-coloring peptides were shown to inhibit cells aggregation and disturb the epithelial morphology and were able to stimulate invasion cells expressing E-colorings. This suggests that E-colorings function are omitted and this inhibition boosts cancer invasion. And that's all. Thank you.
Thank you, Javier. I'm Marta Garcia and I'm going to present the last part of our project, which is about the process of cellular infiltration of tumor and metastasis. The role of tumor infiltration by leukocytes in tumor growth and development is complex. Although TIMs, which is tumor-associated macrophages, have tumoricidal activity and can stimulate anti-tumor T cells, tumor cells can block inflammatory cell infiltration and the function of infiltrating cells. Tumor-derived molecules can regulate the expansion of myeloid progenitors, the mobilization from the marrow, chemoattraction to tumors, and activation, resulting in the promotion of tumor survival, growth, angiogenesis, and metastasis. Numerous studies have shown that activation, and most of them have demonstrated that there is no relationship between immunogenicity, metastatic propensity, and TIM frequency. At first, CD34 move from the marrow into the periphery, in which they differentiate into monocytes, and following invasion to a tissue, mature into macrophages. Macrophage infiltration of brain tumors is regulated by cytokines, growth factors, and enzymes. They can also regulate the function of TAM. However, it has been shown that a low frequency of macrophage infiltration does not guarantee that a tumor will metastasize, as multiple factors are critical. Macrophages present in the extracellular matrix and capsular area of tumors can facilitate tumor invasion and are important during the development of early-stage lesions. Further, the proteolytic enzyme secreted by macrophages facilitates tumor invasion and extravasitation. Indeed, macrophage infiltration has been found to inversely correlate with microvessel density and mitotic index. TIMs also express a number of factors that stimulate tumor cell proliferation and survival and support angiogenesis. Myeloid-derived suppressor cells have also been identified in the circulation and infiltrating tumors.
can inhibit T cell number and function, and the mechanisms are immunosuppressive and are diverse, including suppression of immunosuppressive cytokines. Granulocyte macrophage has been directly associated with them, which can be reversed by blocking antibodies. Vascular endothelial growth factor has also been directly linked with tumor progression. They can suppress tumor immunity and neutralize antibodies. To conclude, I have to say that recently it has been reported that administration of sunitinib, a receptor 2 kinase inhibitor, resulted in a significant increase in MGSC with patients with metastatic renal cell carcinoma. These observations suggest that the regulation of infiltrating myeloid cells has the potential to control the growth of primary tumors and metastases. Well, and this is the end of our